Find your passion, and then once you identify it, go all in for it, no second thought. Being a scientist means that you have the opportunity of really changing how people understand the world.
Dr. Peng Zou fell in love with science at an early age. He loved seeing how chemistry, biology and physics all work together. Now he's combining all three to discover more about molecules in living cells. In this Season 1 bonus episode of Bringing Chemistry to Life, we speak with another member of the Chemical and Engineering News' 2019 Talented 12 about their work and trends in their field. I'm your host, Paolo Braiuca, Senior Manager of Global Market Development at Thermo Fisher Scientific. We began by asking Dr. Zou about the beginning of his interdisciplinary scientific career.
Well, in college, I still got interested in physics, chemistry, and tried to use physics and chemistry as a tool to understand the biological system. Well, I participated in a laboratory research in the structural biology lab, where we were trying to solve the structure of a viral protease, which is key to curing the SARS coronavirus disease, which happened in 2003. We started with the biochemistry to try to purify the protein and use nuclear magnetic resonance, the NMR, to solve the protein structure. And to me, it's a perfect example of bringing together the physical instrument, the chemical techniques, to understand important biological question. At that time, I realized that it's important to understand the biological molecules, its behavior, its structure. But what we are doing really is just in the test tubes. To understand the behavior of the biomolecules, be important to understand it in the context of the living cell, of the live cell environment. So when it comes to grad school, I started looking around different labs, working on the interface of chemistry and biology, particularly in the context of the cell biology.
How was on a personal level, being in such a, multidimensional and multicultural environment as I imagine the MIT is? What did this experience give you from a personal level, even beyond the professional level?
It's a great experience. It feels wonderful. I got to meet with people from all over the world. In China, I have friends all over the country. But in the US, got to meet with the people in other Asian countries, from Europe, and nowadays we are very good friends. I think that's one thing really fascinating about science, being a scientist, because we have set up collaborations really worldwide, and we attend international conferences. And I have friends in Korea, in Thailand, in the UK, in Switzerland. So that really tells me that doing science is an international business, and it's really good to have this international collaboration.
And you said it at the beginning, you were fascinated from the start about the multifaceted aspects that chemistry can put into science, right? Into finding how complex biological systems work, really. And that's what has been your interest since early on. And in fact, what you do is extremely multidisciplinary. And in a way, I would struggle to define it as chemistry. It's definitely more than that. How do you define yourself? Are you more of a biochemist, or what is the definition you give to yourself and your team?
I would like to define myself as a chemical biologist or biological chemist. I work at the interface of chemistry and biology, but I was trained as a chemist, and I'm used to the term describe things in terms of the molecular structure and how structure gives rise to dysfunction. As biological chemists, I'm trying to develop chemical tools to help understand biology. And in my laboratory right now, we are putting together physical instrumentation, some optical engineering, together with chemical tool development. So we are really tool developers at heart and trying to solve interesting biological problems by designing new techniques. And one of the techniques that we use frequently in the lab is fluorescence imaging, because optical technique is really can really allow us to observe cellular activity with a very high precision and high spatial and temporal resolution.
But let's jump into some of the details of what you do in terms of research. So you said that basically you focus on developing chemical tools to investigate biological mechanisms. Reading some of your papers, it seems to me that a lot of these chemical tools are relying on labeling or tagging some of the macromolecules with something that can be measured. Maybe that's where we should start. Am I correct? You're using some sort of enzymatic reactions to attach labels to some of these macromolecules?
Overall, my research program focuses on developing chemical tools for neuroscience. And I got interested in neuroscience early on, I think as early as in high school. I remember the visual system requires a retinal. That lecture really impressed me about how chemistry can help us understand the nervous system, how it works. And the nervous system is really amazing because in our brain, we have over 80 billion neurons. Each one of them has a highly polarized and highly branched structure, which can form over close to 1,000 connections with other neurons. So this is a really complicated system. And in terms of the signaling or cellular activity, that can happen across multiple time scales. At the millisecond time scale, we have these electrical impulses called action potentials going on all the time in the neurons. And then at the hours or days time scale, biomolecules can be synthesized, degraded, or modified. And this can give rise to a plastic change of the nervous system, which ultimately gives rise to learning and memory. And we are interested in understanding the nervous system at very diverse length scales and temporal scales. For example, at the longer time scale for the biomolecules, we are interested in understanding how the localization and the modification of proteins and nucleic acid is involved in the neural function. The traditional way of trying to understand the localization of biomolecules is to take a sample and take it apart and try to run ultracentrifugation or some other physical isolation method to isolate the region of interest, for example, some certain organelles, the nucleus or the mitochondria. And then you can run biochemistry analysis to understand its content, to analyze its protein or nucleic acid content. But this type of separation is not ideal because you tend to lose things during the hour-long isolation, which is laborious and error-prone. You may also introduce some contaminants. So what we are trying to do is to replace this physical separation with a chemical reaction that occur inside the living cells. And just using chemistry may not be enough because it's difficult to control where the chemistry might happen inside the living cell. So the way we try to solve the problem is to introduce a handle, introducing an enzyme to trigger the reaction. The advantage of using the enzyme is that it can be genetically encoded and be genetically targeted to specific cellular compartments. And then we can use this spatially restricted chemical reaction to label biomolecules in close proximity to the enzyme. We can label proteins and nucleic acids, including RNA and DNAs.
I have a couple of questions. The first one is, how do you ensure that the enzyme that catalyzes your reaction that is used for the tagging or the labeling is really happening just in that compartment and not everywhere else in the cell, so that will skew your analysis, right? How do you do that?
First, we need to target the enzyme specifically to the compartment of interest. And this can be done by putting the protein in the form of a genetic fusion with a short peptide or fusion with other proteins, so that when the cell produces the protein, its own cellular machinery can recognize the specific sequence and move the protein or traffic the protein to its correct target. So that's step number one. And then once the enzyme got targeted there, we need to choose a chemical reaction that is only labeling local proteins. The idea is to use the enzyme to catalyze a reaction to turn a substrate into a highly reactive intermediate. It can react quickly with proteins or RNA molecules nearby, but doesn't have enough time to diffuse further away because it's short lifetime. The best type of this highly reactive intermediate would be to use free radicals. But of course, free radical chemistry is always one of the most challenging chemistry. So we tried several versions of free radical chemistry, and at the end we used the peroxidase mediated reactions. I started this type of research when I was a graduate student with Alice team, but now I'm still but now we are continuing developing other type of enzyme catalyzed or even photocatalyzed reactions to improve the spatial temporal resolution.
We hope you're enjoying this episode of Bringing Chemistry to Life. Stay tuned at the end of the episode for information on how to access content recommendations from our guests, as well as information on how to register for a free Bringing Chemistry to Life t-shirt. And now, back to our conversation. So when you said that you genetically modified the cell to ensure that the enzyme that is going to catalyze the tagging reaction is localized in the right place, is you basically fuse it with a protein or a peptide or with something else that you know is going to be present only in the organelle or in the compartment that you're interested in. Am I right?
Yes, that's correct. So fortunately, in the prior research, people have developed various techniques of targeting a protein tag to some cellular compartments, the nucleus, the endoplasmic reticulum, the mitochondrial matrix, for example. And people often use these type of techniques to target a fluorescent protein to these compartments for imaging purposes, so they can visualize the cellular structure very clearly under the microscope. And fortunately for us, we could take advantage of these decade-long research just to replace fluorescent protein with the enzyme that we use, because they are similar in size. They also have other similar properties there. For example, they are monomers, and they don't require disulfide bonds to fold properly, so they are genetically targetable.
I understand. So I'd like to discuss a bit more about the chemistry that happens. You mentioned the use of peroxidases. So basically what you generate is in situ peroxide-derived radicals. And I suppose these react with some of the amino acid side chains of proteins, or what other groups in the nucleic acids? Can you speak about the chemistry there?
Yes, the first version we used is a engineered peroxidase whose preferred substrate is a phenol substrate. So we conjugated a biotin molecule onto the phenol, and the phenol is really the warhead and got turned into a phenoxyl free radical at the active site of the enzyme. The reaction is triggered by the addition of hydrogen peroxide. There's a heme molecule at the binding pocket of the peroxidase. So following its oxidation by hydrogen peroxide, it can then oxidize phenol substrate into these phenoxyl free radicals. The free radical has a very short lifetime in the aqueous solution, much shorter than 1 millisecond. It doesn't really have enough time to diffuse very far away. It can react with the electron-rich amino acid side chains in the proteins, and most likely with the tyrosine residue, which also has a phenolic structure. And we also found that these phenoxyl free radicals can also react with nucleic acids. And we even did some engineering or changing the structure of the phenol structures. We tried aniline, we tried other substituted phenol or aniline structures to improve its reactivity towards nucleic acid.
Are these tags interfering with the biological functions of the protein or the nucleic acid you're labeling? So do they affect what happens in the cell compartment after the label is attached?
In terms of tagging the enzyme, because it's really a big protein tag, we always have to be very careful when putting on the tag. We need to test whether the tagging with the enzyme would cause any change to the protein function. For the small molecule tags, once we trigger the free radical reaction, we're actually causing some type of oxidative damage, oxidative stress to the cells. And we usually wouldn't wait for too long. So it's like taking a snapshot of the cell. The reaction only occurs in a few minutes, and thereafter we have to quickly kill the cell and do this following the cell lysis, extract the proteins or RNAs for downstream analysis. And because free radical is really doing a lot of damage to the cell, but fortunately this happens within a very short time window, so we could see how the cell was like, how the proteins and nucleic acids are localized.
So this is basically your way of freezing that cell moment in time, and then analyzing what the cell was looking like at that precise moment in time via microscopy or further downstream processing and other types of analysis. And then you build up your sort of temporal map with freezing the cell in a subsequent moment and then putting the things together so that you can have like a sort of view of how the chemical mechanisms inside the cell progress or evolve over time, just taking different snapshots. It's like a series of photographies of the cell status. Am I right?
Oh yes, that's correct. It's like taking several sequential snapshots of the cell.
What you can do with this technique is basically understanding what is where in the cell at a specific moment in time. But you can also, if I understand correctly, follow some other types of mechanisms like transmembrane voltage changes and things like that are important in neurons, and that you can use either changes in chemical equilibrium or in potential equilibrium that can cause some sort of chemical phenomenon that you can observe. What are other things that you can do there?
Well, in terms of observing cellular activities, doing the enzymatic labeling can tell us about how the biomolecules are located and what happens to the modifications or their abundance inside the cell when cells are being stressed or being activated. But this happens at a very slow time scale. At a shorter time scale, at a millisecond time scale, the electrical activities or the membrane potentials can change very quickly, and other metabolites or other signaling molecules can have their concentration varying very quickly within the time scale of seconds. To really understand how those cellular activities happens, we need to have some real-time readout. So the fluorescence imaging is one of the best ways of doing this. But to do that, we first need to convert the signal into an optical readout. And this would be particularly challenging for observing the electrical activities because unlike the calcium or other small molecular ions, you cannot really have a binder that recognizes this chemical entity, which can change its conformation, which ultimately leads to a change in the fluorescence readout. Electric field is something you cannot really easily sense. The only way, it seems, is to put a sensor on the cell membrane that can be responding to the changes in the transmembrane electric field. And somehow this is converted into an optical readout. So in a way, to observe changes in the membrane potential is to try to introduce an electrochromatic sensor, converting the electrical signal into an optical readout. The most classical technique is to use the electrical recording. This can be done either by a multi-electrode array or more precisely by the patch clamp electrophysiology. But these often require putting an electrode in close proximity to the cell, and sometimes often to break the cellular membrane barrier so that the electrode can record the whole cell transmembrane potential precisely. But this is relatively invasive to the cell, and these techniques are often very technically challenging to perform such type of experiment. And also it doesn't easily offer high spatial resolution. Although in terms of sensitivity and temporal resolution, electrical recording are fantastic. They are great. They're still the gold standard used in the field of electrophysiology. But people often realize that you cannot put too many electrodes simultaneously, usually just a couple. And it'd be great if you can use a camera as a way to record the electrical signal. Then every single pixel on your camera can serve as your probe.
Obviously, with a less invasive chromophoric sensor that uses chemistry rather than a physical electrode inserted across the cell membrane, you can actually observe on spatially and temporally how the color changes, right, as the signal passes through the cell, while not altering the physiological state of the cell itself too much.
Right. Traditionally, there are two types of electrochromic sensors. One type is based on just the chemical dyes. People realize there are some chemical dyes that are lipophilic. They can easily stain. They can be used to stain the cellular membrane. And for over half a century, people have been trying to develop brighter and more sensitive dyes that can be used to report the cellular membrane potential. But the problem with the chemical dye is that it cannot be very easily targeted to specific types of cells or specific types of neurons. It can go to all the cells. It can be used to stain neuronal cells or non-neuronal cells, such as glial cells. Cannot be used to distinguish between different neuronal subtypes, for example, excitatory versus inhibitory cells. And if you want to have very good cellular targeting, you can use genetically targetable sensors, for example, those based on fluorescent proteins. And indeed, those are very useful, and you can use genetic targeting to observe membrane potential only in glutamatergic neurons, for example. But you can use these type of genetically coded sensors to observe membrane potential only in excitatory neurons, for example. But the problem with the genetically encoded sensors is that they are based on naturally occurring protein structures. Of course, people are doing lots of engineering, directed evolutions to try to improve their properties, make the fluorescent proteins brighter. But this is often done by just doing mutations in the protein, the structure. And really, this, in terms of the structural diversity, this cannot be compared with the chemical dyes synthesized by organic chemists. So really, the best way might be to bring these two together, to bring the best of chemistry and biology together, so you will have a genetically targetable protein scaffold. And on top of that, we'll put a synthetic small molecule which is very bright and photostable and color tunable, and use that as the fluorescent reporter. So you will have a genetically targetable scaffold and a small molecule fluorescent reporter. We call that a hybrid indicator. And the hybrid indicator is basically one of the focus in my lab. We're using this type of strategy to develop more sensitive and brighter reporter for the membrane potential.
It's an extremely elegant strategy, Peng. So what you're doing is basically using biological tools to target the location in the cell where you're actually introducing chemistry to behave as your sensor and as your observation tool, right? It's incredibly out of the box. It's an amazing tool. Where does the idea come from?
I think the idea started with my grad school, because when we were working on enzymatic protein labeling method, we were already using both the power of chemistry and biology, because using small molecule can really gives you a very high flexibility in terms of what molecules you can use, what kind of functional groups to choose, and even those that's not really occurring in nature. But just using chemistry may not be enough, because these small molecules it doesn't really easily recognize your protein target. So often you would need to use an enzyme to use the enzyme, the specificity and the catalytic efficiency of the enzyme to help the reaction to work. So the enzymatic labeling is really the focus of my graduate work. And now we're just trying to extend this enzymatic labeling. The way we build a hybrid indicator is to use the enzyme to modify a engineered rhodopsin protein with a small molecule. So we call this bioorthogonal engineering.
So you're able to use these approaches in very different ways, but the basic philosophy is the same. And now you have developed a series of tools that allow you to quantify and qualify the content in biomolecules of certain cellular compartments. Then you can visualize certain cellular phenomena like the voltage spikes. What is the next challenge?
In terms of the next challenge, we can now do these type of measurements relatively easily in the living cells, but really try to understand how this is related to the behavior or it would be important to do the labeling in the live animals. And for example, we can now use the hybrid indicators to measure neuronal activities in cultured neurons. And to really use these tools in the live animals would require solving the problems of reagent delivery of the enzyme or the expression of the enzyme in the living animals, which would probably require creating transgenic animals. And the enzymatic labeling of proteins and nucleic acids works fairly well inside the living cells. But then to move them into a brain tissue or the brain of a live animal would also face the challenge of reagent delivery.
It's fascinating. What is the end game? Are we going to be able to create a pretty comprehensive map of what happens inside the cell, and going from visual phenomena that we can measure somehow down to the chemical natures of the different mechanisms that could lead potentially to understanding diseases better or identifying new targets to combat diseases?
Well, I think the progress of neuroscience could benefit from the tools that can offer high precision measurement. And this could be either the physical instrument or the chemical tools. And if we have a good way of observing the cellular activity, both at the millisecond timescale or the abundance and modification of the biomolecules, then it might be easy for us to compare samples from the patient or the normal person, trying to understand the underlying causes of neurodegenerative diseases. For example, right now we are using this optical imaging method to study a cell model for the Alzheimer's disease, and we discovered that the actual potential initiation and its propagation dynamics are being altered in the disease model cell, which might form the basis of drug screening for treating this type of disease.
Peng, as we get to the end of our extremely interesting chat here, there's a question I always ask. So now you're still a young investigator. You have accomplished a lot already, and there's a bright future ahead of you. But if you look back and you stop for a second, what is a piece of advice you would give to someone younger, just starting in their career?
Honestly, I usually would hesitate in terms of giving advice because each one of us is unique. We have our own path, and if something works for one person may not necessarily work for the other. But if I were to say, for example, give advice to a younger me, then I would think maybe the best advice I could think for now is to really to find your passion, and then once you identify it, go all in for it, no second thought. Being a scientist is really a very privileged job. It's not just a job being paid to do something, but then you really got to learn basically throughout your life. Then you must be very passionate about what you are doing. And at the end, you should be proud of what you have done. Being a scientist means that you have the opportunity of really changing how people understand the world, then try to do the most important thing and be really proud and passionate. And once you have your passion, it may be even easier for you to recruit students or talk to other people and try to convince people to join your effort, to join your mission to solve this big problem.
That was Dr. Peng Zou, Assistant Professor of Chemical Biology at Peking University in Beijing, and one of the chemical and engineering news students at 12. Thanks for joining us for this Season 1 bonus episode of Bringing Chemistry to Life, and keep an ear out for more. If you enjoyed this conversation, you're sure to enjoy Dr. Zou's book, video and podcast and other content recommendations. Look in the episode notes for a URL where you can access these recommendations and register for a free Bringing Chemistry to Life t-shirt. This episode was produced by Matt Ferris, Matthew Stock and Emma Jean Weinstein.